medicines, making it among the safest and most effective medicines needed in the health system. We find that it suppresses neurodegeneration in tau transgenic flies. It also has it gets into the brain better than other antiretroviral drugs, and it has less toxic effects to mitochondria than other antiretroviral drugs. So because of this, because we saw across systems this retrotransposon activation in the context of tauopathy, and because this drug is very, very safe, we went ahead and started a phase 2A clinical trial in patients with early Alzheimer's disease. It's a 6-month trial, open label, phase 2A. It's a collaboration between the Barshop Institute at UT Health and the Glenn Biggs Institute. My co-investigators on the trial are Dr. Sullivan, who's a neuropsychologist, and Dr. Musi, who's the director of our Barshop Pepper Center in the Barshop. So we're enrolling patients that are participants that are 50 to 99 years old. They have to have a diagnosis of early Alzheimer's disease. We're looking at safety. We're looking at how well the drug gets into the brain in these participants. We have 2 different assays of target engagement. So we're doing reverse transcriptase assays as well as measuring retrotransposon DNA copy number in blood and CSF from participants, comparing before treatment to after treatment. We're also measuring efficacy based on neurofilament light levels in blood and CSF. So neurofilament light is a protein that increases in the context of many different neurodegenerative disorders. So we're hoping that it will decrease after treatment. And we're also measuring inflammation. As I mentioned, these retrotransposons are very similar to viruses. They're very inflammatory. So we are expecting, or hoping, that inflammation will be reduced after treatment. And then Dr. Sullivan is also is doing neuropsychological assessments before and after treatment. So we have 11 out of 12 participants enrolled so far, so I think we will be finished with this trial within the year. So I'm very excited to see about how that's going to work out. We're not the only ones working in this area. Since we and Josh Schulman also published that transposable elements are activated in the context of tauopathy, other people have become interested in this as a therapeutic target in the context of Alzheimer's disease and related tauopathies. So we have our phase 2 going now at UT Health. There's also a phase 1 at Brown University in patients with Alzheimer's disease. A company called Therapeutic has initiated a phase 2 in patients with progressive supranuclear palsy. This is one of the primary tauopathies that I introduced at the beginning of the talk. Transposon activation has also been reported in the context of ALS. So there's a phase 2 in patients with a particular variant of ALS that's caused by an expansion in the C9orf72 region. There are also phase 1 and phase 3 trials ongoing in ALS, additional ones in ALS. And then there's a phase 2 and phase 1/2 in pediatric patients with Aicardi-Gutierrez syndrome, which is a childhood neurological disorder, but it's also associated with transposon activation. So there's really a lot of interest in targeting this mechanism therapeutically, and I think we're going to hopefully have exciting results within the next few years. All right, I just want to conclude before the panelists come up for our discussion with kind of a bird's eye view of what I think is happening in the context of tauopathy. So I mentioned that tau causes an overstabilization of that actin cytoskeleton. This changes the structure of the nucleus. It changes overall DNA packaging within the nucleus. I didn't talk about this type of small RNA that's important for transposon silencing, but we know that those are depleted and driving retrotransposon activation as well. And then all of these things I also didn't mention cause neurons, which are normally post-mitotic, to reactivate the cell cycle. And we know that the cell cycle activation in terminally differentiated neurons is sufficient to kill those neurons. So when you take a step back, maybe you're all thinking this yourself, like that's a lot of stuff, you know, that's a lot of steps, that's a lot of stuff. And when I look at this schematic, what really sticks out to me is all of the parallels that are happening in tauopathy compared to cancer. So in cancer, it's also a disorder of lack of cell cycle control. So unlike neurons, where you activate the cell cycle and the cells die, in cancer, you activate the cell cycle, they keep cycling, and then they can form a tumor. But it's still loss of cell cycle control. Retrotransposons are also activated in the context of cancer. They're also activated in less developed cell types. So during neurogenesis, for example, retrotransposons become activated. This relaxation of heterochromatin. In cancer, you have more open chromatin. In less developed cell types, you have more open chromatin. And then it's not until those cells start to differentiate into their more mature cell type that you get a tightening over the genome so those cells can specialize. The understabilized nucleoskeleton, these invaginations of the nuclear envelope and blebs of the nuclear envelope that are happening in neurons in patients with Alzheimer's disease, they're also very prominent in various cancers. You see that the nucleus is not a nice sphere, but it has all of these morphological aberrations. And then the overstabilized cytoskeleton also, you can see these actin rods form in less developed cell types. So I think what this whole schematic really reflects is potentially neurons losing control over the program that keeps them in a terminally differentiated neuronal state. So they're losing their identity and drifting back more toward a less differentiated, more plastic cell state. And I'll leave you with that idea and welcome my panelists up to the stage. But I do want to thank a lot of the early transposon work was performed by a postdoc in my lab, Winnie Sun. The transposon people in my lab now are Paul, Camille, and let's see, Elizabeth is working on the double-stranded RNA transposon project. I've listed the funding specifically for the transposon tau work in my lab. We're working on other things in the lab as well. The Owens Family Foundation is funding the clinical trial. The Rainwater Foundation and Tau Consortium funded a lot of the early work that allowed me to get the preliminary data I needed to compete for an RF1 from NINDS. The MD Anderson Consortium has funded a lot of the work in my lab, and I mentioned it provides us with brain tissue. And then the Pepper Center has really provided a lot of support for the clinical trial that's ongoing now. So I will stop there and welcome, I think they're getting formally introduced. Okay.
Thank you, Bess, for that really good talk. That was very clear. I've heard you talk before, so you're still really clear. So I'd like to introduce our two discussants. Constanza Cortez is joining us from the University of Alabama at Birmingham, and she's an assistant professor there in the Department of Cell Developmental and Integrative Biology. Her research focuses on brain aging in general and the role of exercise and exercise-associated circulating factors as novel therapeutic interventions against age-associated neurodegenerative diseases. Our second discussant is Christian Lasagna-Reeves, and he joins us from Indiana University School of Medicine, where he's an associate professor in the Department of Anatomy, Cell Biology, and Physiology, and the Stark Neurosciences Research Program. His work focuses on mechanisms by which tau aggregates to become neurotoxic. So Constanza and Christian, when you come up here, please, and join us. So the two of you actually study different phases of that wonderful line of causality that you just presented as your final slide. And I think I'm trying to ask a question to start this conversation, so it's really going to be a two-part question. What alternative interventions would you suggest, Constanza? And what do you think are the primary inciting mechanisms that actually cause the tau aggregation in the first place, Christian? Why don't you go first?
Of course. So that's a great question, and I think it's one that all of us in this room think about. And I think the way I'm going to phrase my answer is what we were discussing earlier today about this idea of multi-targeting interventions. The field I'm in, which is exercise, I see more so as an adjuvant or a fertilizer for the brain, as we were talking last night, that will provide assistance for more targeted therapeutics like antivirals or anti-inflammation targets. So I think the future of Alzheimer's disease therapy is going to be multiple targets. And also, as Dr. Frost so beautifully presented today, Alzheimer's is many diseases. And so I think we're going to need many therapies for it.
Thanks.
Okay, that's a difficult question, to be honest. I think that it's in the context even like the disease is multifactorial, as Constanza mentioned, and in the context of tau aggregation or mechanism of aggregation, also I believe that there is going to be multiple factors. Like physiologically, yeah, tau is a microtubule stabilizing protein, but you can find it in physiological situations in the nucleus, at the synapse, associated with the mitochondria. So it's not solely one tau, I should say. It's like highly heterogeneous, and there are several conformers, and that it makes even more complicated with the entire aggregation process. So the whole mechanism of aggregation, you're going to have a huge diversity of aggregates. So maybe you're going to have one that will be key for what Bess showed today, triggering this transposable activation, but you're going to have other conformers that are going to be maybe essential for propagation. So which one is the one that we target, or we need a multi-target within the tau aggregation as well? Like, just as an example of the immunotherapy, you want the N-terminal fragment of tau that went to clinical trials without being really successful, so they think maybe it could be a middle region, the epitope that you have to target in order to stop propagation. But that species that is key for propagation will be the key species for what Bess showed today. So maybe we can stop propagation, but we don't stop the transposome activation. So that make things a little bit more complex.
No, that makes sense. And
yeah.
So I was thinking, I mean, there is a large literature in the neurodegenerative field about mitochondrial dysfunction, for example. So what about tau and mitochondria?
Yeah. So work from the lab where I was in a postdoc found that the effects of tau on actin affect mitochondrial fission and fusion, which then affects mitochondrial function. Yeah. I think about mitochondrial changes as a consequence of actin. But
it's something interesting that the majority or the newest interactome studies for tau, the two biggest clusters are mitochondria-related proteins and synaptic-related proteins. So we were thinking how that makes sense if you have the presynaptic proteins at the presynapse and mitochondria when we will be more cytoplasmic. And it could be different species of tau, the one or entities of tau, that one is interacting with mitochondria and being involved in the physiological function of mitochondria. Or it could be this, it hasn't been studied in detail, but there is some research about synaptic mitochondria. So it could be, again, the tau could be playing a role for this transportation of mitochondria to the synapse. That it has been studied more maybe at the basic level, but not in the context of Alzheimer. But definitely something that we work a lot in tau interactomes and how proteins interact with the seed, they aggregate to propagate, and we always observe mitochondria-related proteins that interact and synaptic. So definitely something there. We're not sure exactly what it is, but there is something, not all the ants, but other groups have shown something similar as well.
what do you think about mitochondria from the exercise standpoint, like peripheral mitochondria? Do they affect the brain?
So since, thinking about this idea of circulating factors, there's something in your blood that communicates from the periphery into the brain. Mitochondria actually release pieces of DNA, and there's all this research in the cardiovascular field where the heart, before it's going to have a stroke, it releases essentially signals for help that are pieces of mitochondria. And so there's definitely a lot here that we don't know. And to bring it back to what you guys were saying and about therapeutics, I think they're actually going to be different if we're thinking about prevention versus intervention.
Yep.
Right? And so early stages of disease will probably have to be treated differently than late stages of disease and for prevention of disease in the first place. And so thinking about all of those stages as well is where what you guys are doing is going to be so useful to try and identify those stages, like you laid out that timeline, is where you're intervening and what is that intervention going to look like.
Mm-hmm.
What are some of these exercise-related positive factors that might be interventions that you're studying?
Yeah, so it turns out that whenever we exercise, most of our metabolic tissues, which is all of them really, secrete things into the blood. So far we've identified multiple that originate in skeletal muscle, multiple that originate in liver, and then, like I mentioned, the heart. And so there's beautiful research out of Saul Villeda, I believe he was here recently. And so he's the one that's pioneering this idea that if you take the blood from a runner and put it in a sedentary individual, the body responds as if it was running. And so that's how we're approaching this idea of targeting these factors, and can we use them to develop these essentially brain targeting therapeutics to provide those benefits without having to get on a treadmill, because a lot of the populations we're trying to help can't.
Well, I'm wondering if we should open this up to some questions from either the audience or, so let's do the audience first, and Dr. Truchard. We're going to give you a microphone.
When you showed the picture of the amyloid beta and then the picture of tau, they were different. So how can you explain the connection between what's happened with the amyloid beta and the tau, and the fact that different regions of the brain?
Great, yes, great question. So tau clearly does not need amyloid beta to aggregate. You see all of these different tauopathies where you have tau aggregates and there's no amyloid beta. So I think that there are I don't know what causes tau to aggregate in the first place specifically. I mean, clearly it becomes aberrantly modified and starts to aggregate. But I think the initiator of tau aggregation can be different. And that's going to change which cells start to have tau aggregation. In the context of amyloid beta and tau, I mean, tau, as we were talking about earlier, it starts to aggregate in a region of the human brain called the locus ceruleus in humans in our teens. And we don't have any amyloid beta there. And it's not until you start to have, in the context of Alzheimer's disease, that amyloid beta deposition elsewhere in the brain that it seems like the tau begins to spread out of the locus ceruleus. Now, if that's something that amyloid beta is promoting, I don't know. I think it's a hard question to answer. Does anyone have a better answer?
I agree.
Sure.
Yeah, in the context of Alzheimer, yeah, you have amyloid, but how Bess mentioned, there are different other triggers to that produce this aggregation of tau, like in chronic trauma-encephalopathy, any trauma exposed to environmental contamination like toxins, so overall oxidative stress, or in frontotemporal dementia, there are mutations that will facilitate this accumulation and aggregation that you don't have in patients with Alzheimer's. So again, that's the big question, to know exactly what is the specific event that triggered tau aggregation and this association with amyloid, but I think it has been shown that there are several factors that independently can start this event, but how and why, I don't think that is clear. But I think it's a good point, like when you show the MRI of amyloid and tau, they are not perfectly overlap, so there are areas that you will have this amyloid tau, but there are areas only amyloid, but I don't know what will be the reason of that, to be honest. Mm-hmm. Yeah, so that, again, it will be great to be able to answer it perfect, but I don't know if we know the answer by now, how or what triggered that event of this association between amyloid and tau.
David.
Dr. Frost, thank you for the great talk. Really appreciate it. Dr. Cortes, you mentioned this exercising muscle releasing factors, and the brain also has dramatic increase in activity with certain kinds of exertions. Is there any evidence that the brain itself can excrete or release factors that help its own repair?
Yeah, so in response to exercise, BDNF, brain-derived neurotrophic factor, is significantly increased in multiple brain regions, but especially in the hippocampus. And so we don't know exactly what the signal is, right? Is it physical, is it mechanic, or is it some sort of peripheral signal that tells the hippocampus to turn on BDNF? But we know that expression of BDNF is required for the brain benefits of exercise. And that's one of the big areas of investigation in the field of neurocognitive exercise medicine is where is this BDNF coming from? Because another thing that we sometimes don't think about is, we talked a lot about neurons today, but astrocyte and microglia are there too, and they also make BDNF. So, these conversations can also be disrupted in the presence of tau, and how do they contribute to essentially lowering that threshold where the brain can resist the tau load before it crashes into disease and pathology is something that we're trying to figure out.
We have a few questions from the outside, and I think, Bill, you're going to...
Well, I had, if I
Your
can
own?
ask my own.
You go ahead, ask your before you become
Yeah, I could ask my own.
ambassador.
And then we do have from the virtual audience. It was interesting, Bess, that you mentioned about the locus coeruleus and sort of the Big Bang or the origin of this. So made me wonder, because of course synucleinopathies also in a way begin in the pons, right, with REM sleep behavior disorder. Might not be the locus coeruleus, but what's with the pons, if that's where these things begin?
What's with the pons? I don't know. I don't know. What's going on there? Why are these proteins aggregating there first?
That's a good question.
I don't think it's known.
I don't think that is
Yeah.
known. Yeah, even in different tauopathies, where tau accumulate is different. I think also there are tau accumulation in PSP in the pons. There is not an explanation, or we don't know exactly why this happened in one disease and in another disease in another brain region. So
Maybe dreaming too much or too little?
Maybe.
We have a question from Luba Azersky, postdoc here at UT Austin. Wonders, have you seen, and this is the best, have you seen a differential impact on tau isoforms with therapy?
With therapy. We have not investigated the effects of 3TC on different tau isoforms. This transposon activation is downstream of tau. It's not something that's driving differential tau phosphorylation or differential tau splicing. So we don't think it's going to affect tau itself, but the issue of the tau isoforms is a good question. So tau is differentially spliced, so you can have a different number of microtubule binding domains, you can have a different number of the N-terminal domains, and we have not looked at whether, for example, it's 4 repeat versus 3 microtubule repeats that drive transposon activation. Actually, I guess we kind of have. Okay, so in flies, our model is a 4 microtubule binding repeat model in flies. In the human Alzheimer's disease brain, they have, in neurofibrillary tangles, for example, it's a pretty good ratio of the human brain has 4 repeat and 3 repeat tau. So this transposon activation we're seeing in the human brain seems like it's relevant to all of the isoforms that are present. It wouldn't be restricted to a certain splice isoform of tau. Yeah, I think that's important. That's why it's nice to go from flies to mice, which is actually a different tau mutation than what we're using in the flies, and then humans, which is a sporadic tauopathy with no specific, no mutations in the tau gene. And if you see the same thing across systems in the context of different mutations, it seems like it's maybe more relevant broadly for tauopathy as opposed to being something that just happens with the R406W mutation, for example.
can I ask a question?
Yeah.
So you observed that in PSP cases, that is a 4R repeat, Alzheimer.
Yeah.
Have you evaluated that in peak disease, for instance?
No, we've only looked at Alzheimer's disease and progressive supranuclear palsy. We don't have brain tissue. We haven't analyzed Pick's disease. Yeah, and then when we're looking at transposon activation in humans, we're analyzing RNA levels, which doesn't reflect everything that transposons do, and then we're looking at novel transposon jumping events. That's only in Alzheimer's disease patients that we've done that, not in not progressive supranuclear palsy. So, but we have a grant submitted for PSP for that project. So, yeah.
We have one question over there. And I think then after this question, we're going to have to wrap it up. So.
Thanks, Bess. So when transposable elements jump into the genome, where do they land, and are they expressed? What are they?
Yes, great question. So for the human data that we have, the nanopore long-read sequencing, we see that most of the transposable elements are jumping into intergenic regions, and then when they jump into a protein coding gene, it's usually into an intron. So that makes you think, if these transposons are jumping into intergenic regions, maybe it doesn't really matter. So one of the things that we want to do is see where they're jumping in, and then how it affects the type of sequencing that we've done. We can see where it jumps in, and then we can look at DNA methylation changes close by to see if it's changing the epigenetics of nearby genes. So that's one of the things that we're working on. But yeah, I think it's a really good question. Yes, there's as definite as you can be in science. There are new somatic retrotransposition events in normal human brains, in the early Alzheimer's disease brains, and at late stages of Alzheimer's. We can detect it in all of the situations. But does it really matter in terms of what they're jumping into? I don't know if it does yet. I know that the drug suppresses neurotoxicity, but is it because it's reducing that episomal DNA that's not actually inserted? Maybe. Yeah, we don't know yet, but that's a great question.
Maybe changes in your inflammatory markers, if that's the prominent feature, will be a clue.
One other thing, whenever the line 1, for example, whenever it tries to insert, if it fails to insert into the genome, it can cause DNA double-strand breaks. So even if you don't get a legitimate insertion event, you can have DNA damage where that transposon has tried to jump into. So, yeah.
Well, I think our time is about out. Am I right? Yes. So I want to thank the three of you, Drs. Frost, Cortez, and Lasagna Reeves. Can we give them a hand? And I just want to put in a plug for UT Austin. We do have some basic research here in Dr. John Pierce's lab in the College of Natural Sciences using, somebody gave the hook 'em horns, the scene, using C. elegans as a model system. And then there's, of course, the UT Austin Comprehensive Memory Center in the Neurology Department in Dell Medical School doing clinical work. So those are 2 points of contact on this campus that you might wish to interact with. And of course, San Antonio's, an hour and a half away. So if you have a good idea, you know who to call. So thanks for being with us, and be on the lookout for our next Oskar Fischer lecture in a few months. And we hope to see you then. Thanks very much. Good job.